How's it going, everybody? Today's topic is the cell cycle. The cell cycle is just a fancy word for saying the life cycle of our cell. There's several parts to the cell cycle, but it's pretty basic. First, though, before we start talking about the parts of the cell cycle, we should probably talk about what the purpose of a cell's life is. So, all cells have two purposes. Purpose number one of a cell is to do their job. What that means is if they're a heart cell, they should be a heart cell. If they're a liver cell, they should probably be a darn good liver cell. If they're a blood cell, they need to be a blood cell. Their goal is to be the best they can be at their job. Their second goal should be to reproduce. So we're going to see that this occurs at the end of their life cycle, and that's kind of like the mecca of their life. If they reproduce, then they've reached their goal. They are a good cell. Like I said, there's certain parts to the cell cycle, though. So the cell cycle has several stages. The first stage is something called G1 or gap 1. The second stage is something called synthesis or S stage. The third stage is something called gap 2 or G2. And the final stage is going to be something called mitosis or M. Alright, there's also going to be an offshoot of the cell cycle that could occur at any point in the cell's life, but that offshoot is called G0 or gap 0, not go. It says gap 0. So I'll put a little thing through it so you know that that's 0. So this is a life cycle just like you and I have a life cycle, just like humans have a life cycle, where we go from infants to toddlers to tweeny boppers to teens to young adults to old people, adults to old people to, death. Just like that, cells grow through different periods of their lives, where they go from being a young cell to being an old cell. And this is the cycle. What we're going to see is that here, this part, the cell cycle part, is when the cell is focused on its goal of reproduction. Over here, when the cell's in what we call gap 0, the cell's just going to be focused on doing its job. So it's just going to be focused on that first goal instead of the second goal of reproduction. Alright, so here in gap 1, the first stage of our cell's life, when it's just a young little strapping cell, there's one goal and one goal only. So if you see here, actually there's two goals, I lied, there's two goals. But if you see here, this is our young little cell membranes in white out there. This is the nucleus. You see the nuclear membrane in white. There's the DNA in the middle of the cell. And here, these guys, those stars are representing organelles. So in the first stage of its life cycle, during gap 1, the cell is going to do two things. First of all, it's going to grow. The reason why it's going to grow is the same reason why if you were going to cut a piece of cake for 40 people, you'd want to get a big cake. If we keep cutting our cell in half when it divides at the end of its life cycle, then it's not going to be very big for long. It's going to keep cutting in half, and so if one cell splits, its offspring will be half its original size. And if it splits again, it will be a fourth the original size. That's a problem. So our cell has to grow so that when it makes new cells, they're going to be big enough. The second thing that's going to happen is that our organelles are going to be copied. So our cell is going to copy its organelles so that now it has an extra copy. The whole point of having an extra copy is so that when it makes a new cell, that new cell is going to have its own organelles. So you can see growth and organelle replication are big, most important things in this phase. After the cell's completed gap 1, it moves on to something called synthesis. Synthesis is simply put DNA replication. I'll talk more in depth about DNA replication at the end of this little presentation, but what we know is that our DNA is going to be completely copied. The whole purpose of the DNA being copied is so that when we make a new cell, it's going to have the exact same instructions as the original cell. And it can't just cut, the original cell can't just cut its instructions in half. It can't just cut its DNA in half, because then the new cell would only have half the instructions. That would be kind of like if I invited you to play Sorry with me, the game Sorry. And I said, here you go, here's half the instructions. You're not going to know what's going on. You're not going to know how the game works or what the rules are. Right? Sorry's maybe not the greatest choice. That game's it's not the funnest game in the world. But you get what I'm saying. So S is all done for DNA replication. As you can see, both G1 and S have fully been focused on the cell preparing for division. First in G1 by growing and making new organelles, and then in S by making new DNA. After the cell completes DNA replication, it moves into gap 2, the third phase of the cell cycle. During gap 2, the same exact goal of gap 1 is occurring. The cell grows in preparation for cell division so that the new cell is going to be big enough to function. All right, this one's pretty simple. Easy way to remember it, gap, grow. In both gap 1 and gap 2, the cell's growing. GG. So the G's are growth. G's growth. The final stage of the cell cycle then is the actual part where the cell really is getting ready for division. It's called mitosis. So cell division, or at least nuclear division and DNA division, occurs during the mitosis. What I mean by DNA division is I mean that we're going to split the DNA so that both of the new cells are going to have the DNA. At the end of mitosis, a process called cytokinesis occurs. Cytokinesis is where the cell actually splits into two separate cells. And at that point, the whole entire cell cycle would begin again. If you remember, though, I said that the cell also has another offshoot where it's not really in the cell cycle. That's called gap 0. In gap 0, the cell's just doing its job. It's just focused on its first goal where it does its job, unlike where it's in the cell cycle where it's focused on division. So here, our cell isn't going to be dividing, isn't going to be doing anything like that. It's just going to be doing its job. And the reason why we would have a cell that's in gap 0 is that if you think about it, we don't always need new cells. So you need new cells when your cells get injured or they die or you want to grow or they're damaged. But you don't always need those new cells. If your cells were constantly dividing, that would actually end up being a problem. So when cells don't want to divide, they just focus on doing their job and they spend all their energy doing their job. We say they're in their gap 0 phase. Fortunately for us, throughout this whole entire process of the cell cycle, our bodies have specific checkpoints where they check the cell and make sure that everything's okay. So at the end of G1 and at the end of G2, our body stops the cell from moving on to the next step, whether it be synthesis or mitosis, and makes sure that everything's intact. Is it big enough? Is the cell copied its organelles? Has the cell copied its DNA properly? And if the answer is no to any of those questions, one or two things will happen. First, either our cell will begin whatever the previous stage was. It will start growing some more. If it needs to get bigger, it will recopy its organelles if it needs to make new organelles. Or it will fix the errors in its DNA if it copies the DNA wrong. However, sometimes the problem's so big that our cells can't be fixed. Our body has a way to deal with that. What it does is it says, all right, cell, you're going to read this little piece of DNA here, this little gene, and you're going to get a specific instruction. And that specific instruction says, hey cell, explode yourself. Kill yourself off. That's right, the cell will commit what we call cell suicide or apoptosis. And the cell, being the willing little servant it is, will say, all right, I'm willing to sacrifice myself for the good of my body. And it will explode itself. Kaboom. The end of that poor little cell. All right, and that's a way to prevent our cells from being corrupted and then going into mitosis and making offspring that are also corrupted. At the end of the cell cycle, we'll have started with one parent cell, and we'll end up with two identical daughter cells. They're going to be identical to each other, and they're going to be identical to the parent. And yes, they are called daughter cells. There's no son cells. Whatever, though. I guess we'll just deal with that. And that right there is the cell cycle. So in conclusion, you've got 4 major stages where the cell's focused on preparing for cell division. Gap 1, where it grows and copies the organelles. Synthesis, or S, where it copies its DNA. Gap 2, where the cell grows. And mitosis, where the DNA divides and it prepares for cytokinesis or cell division. And then there's also 2 checkpoints in there to protect our cells from making new corrupt cells, as well as a phase called gap 0, where the cell is focused solely on doing whatever its assigned task is.